Hello, everybody, and thank you for joining us today. I'm Birgit Washburn, editor at Wiley, and I'm here to introduce this webinar entitled Single-Cell Injection, Deposition and Aspiration, Force-Controlled Manipulation with FluidFM. Today's webinar is brought to you by NanoSurf. And we are pleased to have Edward Nelson from NanoSurf and Oren Guillaume-Jonty from ETH Zurich joining us here today as our presenters. Edward Nelson joined NanoSurf as a support scientist in 2015. He has more than 15 years of experience as an experimental nanobiophysics with a background in single-cell manipulation. He earned his PhD in physics from the University of Rochester and was a postdoctoral fellow at the University of Notre Dame, where his interests included single-cell optical tweezer lithography, synthetic biology, and single-molecule atomic force microscopy. Oren Guillaume-Jonty is a senior scientist in the Department of Biology at the ETH Zurich in the laboratory of Professor Julia Vorholt. Over the last 8 years, she has been driving the development of the FluidFM technology for single-cell biology. She has established different approaches that enable selected perturbation and analysis of living individual cells, including intracellular fluid injection and the sampling of cellular biopsies of ex situ molecular analysis. Just before we get started with today's event, let me just remind everybody that this is an interactive web seminar, and so you can ask questions at any time during the presentation by clicking into the questions box. Now, only your presenter will be able to see your questions, so don't be too shy about asking them. Questions will be answered during the Q&A session at the end of this presentation as time permits. Now, if Edward and Oren aren't able to get to your question today during this live broadcast, then they will do this best to follow up with you later offline. So, without further ado, let me turn the floor over to Edward, who will begin our webinar today. Okay, Edward, it's over to you.
Thank you for the introduction. As you can see, the title of this webinar is Single-Cell Injection, Deposition and Aspiration. I'm going to spend a couple minutes giving you a little bit of background about the FluidFM microfluidic system. I'm going to also describe a little bit about single-cell and force control using the FluidFM system and how FluidFM can achieve this. So I hope by the end you'll have a better appreciation for what FluidFM can do and how it can help you with single-cell analysis. So first question is, why single-cell analysis? Well, cellular populations are heterogeneous, so the ensemble average can only reflect dominant biological mechanisms. This can hide effects from small yet important subpopulations. So, for example, antibiotic resistance persister cells, for example, or dormant cells. In populations containing several phenotypes, the average may not correctly describe both subpopulations. Also, bulk measurements do not account for cell-cell signaling or mechanobiology between cells. So, in summary, single-cell analysis really informs how a subpopulation of cells interact in a complex structure such as organs, tissues, and biofilms. Second question is, why force control? And short answer is, has everything to do with stress on a cell and trying to minimize that stress. So external stress can affect cellular function. This can be chemical, thermal, or mechanical stresses. Now, there's a classic cellular stress response. This could be to try to repair itself or to induce cell repair. The cell can try to adapt to the new environment. It may reorganize or shut down components that are unnecessary to the new environment. This is called autophagy. Or it can trigger apoptosis, which is like a programmed cell death. So understanding, and this could either be minimizing or quantifying the external stress, is fundamental to an analytical single-cell biology. So how does the FluidFM system work, or how does it have force control? Well, FluidFM is a microfluidic system, and it has a probe at the end of a microfluidic channel, and the probe itself is a cantilevered spring. We can detect deflections in this microfluidic channel with the use of a laser, and deflections in the probe are proportional to the force. So as we push down on a cell or on a surface, we can see the microfluidic channel deflect, and from there deduce what the force on the cell or sample is. what does allowing force allow us to do, and how can you quantify stress? Well, because we can measure the force and also distance with the microfluidic probe, we can measure both. We can quantify both the cellular deformation and stress. So here's two images or two plots. On the left is a yeast cell. On the right is a HeLa cell on fibronectin. The blue curve there shows the deformation on the x-axis and force on the y-axis. So interesting to note that the mechanics of both cells are very, very different. So on the left, you can see that the blue curve looks very different from the one on the right, and it has a little bit to do with form. So yeast is a long and thin cell. HeLa is globular and squishy a little bit. And so the biomechanics between the cell and the surface have a lot to do also with the biochemistry in the system. The last thing that the system, the last part of the FluidFM system is it's integrated with a microfluidic control system. Now, this is computer controlled. It gives you a large range of positive and negative pressures, so you can pick up cells or deposit them. It's fast. As you can see on the video on the right, we have a fluorescent dye inside the microfluidic channel, and there's pulses that are pushing this dye out through the aperture in the front of the microfluidic channel. Everything together makes the FluidFM a miniature pipette tip. So what can we do, or what good is force-sensitive microfluidic system? Well, for one, we can do force-controlled injection and extraction. And I won't spend a lot of time on this part, but because the second half of the talk will be extensively on injection and extraction. But with force control, we know exactly where in the cell an injection aperture is located. So again, here's a force displacement curve. Like before, the x-axis is the distance above the surface of the cell, and on the y-axis is force. At position 1, we make contact with the outer cell membrane. If we apply a little bit more force, we can eventually break through the upper plasma membrane at position 2. Continuing to apply a little more force, we go through the upper nuclear envelope at position 3, and so forth until we reach the actual surface. So we can push all the way through this cell. So this is important if we want to decide where in a cell we like to inject or extract material. So for example, if you wanted to inject into the nucleus, we would apply between 75 and 175 nanonewtons of force. So this is between positions 3 and 4 in this graph. If we wanted to extract, for example, material from the cytoplasm, then we only need to apply between 25 and no more than 75 nanonewtons, so between positions 2 and 3. So being able to measure how much force we apply to a cell during an injection or extraction experiment means that we can be more confident that the material goes where we want it to. Now, the last component of the FluidFM system is an integrated nanomanipulator. So this allows us to address any cell on our sample. It has a large range, so it can cover tissue samples. It has high precision, so we can address specific regions within a larger cell, or we can even target single bacteria. On the video on the right, this shows the feature there has a 100 micron diameter, so that gives you the idea of the precision there with the nanomanipulator. That's essentially a FluidFM system in a nutshell. So let me get into some of the applications that have been done. One is to study the heterogeneous population of single-cell organisms. This is research out of the UK and the Netherlands. So waterborne protozoa, these are oocytes that have been implicated in various water treatment contamination incidences. In the typical measurement protocol, as you can see here in the 3 images, on the one on the left, we can see the end of the microfluidic aperture there and a target cell. With a negative pressure, the cell is drawn into the aperture, and in the second image in the middle, the cell is measured. So whatever measurement you want to take with it. Finally, with a positive pressure, on the third image, the cell is released. And we can repeat that for as many cells as we want. That's the benefit of the system. We can do serial measurements on a single sample in a short amount of time. In the results of this experiment showed that within a single population, there were 2 subpopulations of organism. We have a pathogenic and a benign cells within this population, and they differ in both the size and in the stiffness. So these are two measurements that were taken with the FluidFM system, a force measurement and a size measurement. In the image on the top image, or the graph on the top, you see the circles and the squares representing the two types of cells. A little bit more interesting result is if you look at the difference between treated, so these are non-infectious oocytes versus the infectious oocytes, there is no difference in the size of these cells. So using the height of the cells alone is not enough to establish if it's been treated or not treated. But if you look at the stiffness, so the elasticity or the force on the cell, and the graph on the right, we see that there is a large difference between the heat-treated and the untreated cells, and there's also a difference between the freeze-thawed treated cells and the heat-treated cells. The asterisks above those box plots indicate that there's a statistically significant difference between the data sets there. So the next application I'd like to get into is using the FluidFM microfluidic system itself as a force sensor. So why is it different from traditional techniques? So in traditional cell adhesion measurements, some of them aren't quantitative at all, such as washing or laminar flow measurements. Some are not reproducible, so either spinning disk or centrifugation are not repeatable. With the FluidFM system, the force on each cell is measured directly and with high resolution. Once we do that, a more detailed picture emerges of the cell-substrate interaction. So in the video on the right, we see a typical cell adhesion measurement. First, we select a cell using the crosshair. We bring the microfluidic cantilever to this cell and approach. We apply under pressure, and then the sample is withdrawn. And when this happens, we can measure now the force between the cell and the substrate. And what we see there in the graph is the force displacement curves that I've been showing before. So cell-substrate interaction measurements, these are of great importance to researchers working with engineered surfaces. So for example, medical devices. One group used FluidFM to measure the interaction force between single cells and different engineered substrates. So notice the diagram on the left, the FluidFM probe on top of a cell. We have the force sensing laser there. The substrates all varied from a flat surface to surfaces with periodic grooves of different depths. Now, FluidFM could quantify the force and found statistically significant differences between the different substrate designs. The plot that says bleb there, that's something that was applied with blebistatin. That's a myosin inhibitor, so that reduces adhesion. So you notice that the adhesion force is much greater than for the 100 nanometer grooves. Another thing we can do with the FluidFM system is measure cell-cell interaction forces. This is a unique measurement, and there's no really alternative in physiological conditions. So how cells interact with substrate and interact with each other in a monolayer, this was research done in Germany, and what they did is they looked at endothelial cells. These are the ones the ones on the left. I'm looking at the graph on the right and the plot on the left. And L929, these are fibroblast cells. So these are connective tissue cells. And what they found for the endothelial cells, there's large differences between individual cells and cells in a monolayer. But if you look at the fibroblast cells on the right, there was no significant difference between the two. That's interesting. Just moving on. Next topic, and this is maybe the most important part of the talk, is deposition. So in this case, we're going to fill the microfluidic channel with particles, small particles, or viruses in this case. And the schematic on the left shows essentially the protocol. You can see the virus particles inside the microfluidic channel. There's a laser above it. for the force deflection or force detection. The microfluidic system is brought in contact with the cell. This does two things. With the force control, we can ensure that we don't damage the cell, reduce its stress. And another benefit is if we bring it into contact, we block the viruses from going to other cells. So we know we can target a single cell at once. As we lift the FluidFM microfluidic, we can now see the virus particles. In this case, they're dyed with RFP. They have an RFP marker. And in the fourth one, bottom right, we see that the virus has infected the cell. The video on the right shows these virus particles inside the microfluidic channel. They're RFP labeled as they exit the FluidFM probe. So a typical protocol or a typical measurement would look like this. First, you have an image of a cell, of a colony of cells. We select the cell using the cursor. The probe is automatically positioned above the cell and approaches the cell using force control. We can deposit the virus particles, and then we retract the microfluidic cantilever. And at that point now we can count the number of virus particles on the cell, and in this case there are 4. Because the FluidFM system is integrated with an optical microscope, we can track the infection process with fluorescence. In this case, the virus particles were designed to transfect GFP in the cells. After 7 hours, we see a little bit of fluorescence due to the GFP. Let's move on. And after 11.5 hours, the intensity increases. So we know there's producing more GFP, green fluorescent protein. And at the same time, after 11.5 hours, we start to see on the RFP channel, we start to see more virus particles within the cell. So the cell is producing more viruses. So we can put large particles into the FluidFM probe, or we can put small particles or chemicals inside the FluidFM probe. In this slide, I'd like to describe an experiment that was done to locally stimulate neuronal cells using glutamate. On the left, you see a schematic of the FluidFM system. You have the microfluidic channel, the laser for force control, a neuronal cell on a microelectrode array. What they're going to do is just target a single cell and deposit a certain amount of glutamate on there. So what they found was the minimum amount of glutamate necessary to activate the cell, and they found around 8, almost 9 micromolar of glutamate. They also determined how far or what the minimum distance away from the cell they needed to be in order to target just a single cell. That's the bottom right figure there. So what they did is they targeted a single cell, and glutamate activates gated ion channels and G protein coupled receptors in mammalian cells. So every time they activated the cell with the glutamate, those are the red spikes you see there, they saw a corresponding voltage spike in the potential. So this could be an alternative to something like a caged neurotransmitter like caged glutamate or caged serotonin. The difference here is that we do not require a light source for photoactivation. So in a cage system, you need to apply light to break the photo label. And it could be used with any compound. So it doesn't require a photo label precursor for activation with the FluidFM system. Now, because the FluidFM probe is a microfluidic probe, we can actually use the probe itself to measure the action potential or voltage-gated receptors directly. Added benefit is that it's force sensitive. So if there's any physical changes to the cell during a patch, like if the cell contracts or swells, the probe will react, and we can maintain the ohmic seal on the patch. We can also measure physical changes to cells during voltage clamp pulses. So if a cell also reacts to a voltage pulse, then we can see that happen. So one of the obvious targets for this is a cardiomyocyte cell. So it's like a muscle cell, right? So during a contraction, in this figure, we see that there's a force on the cantilever, on the probe, due to a contraction in the cell, in the cardiomyocyte. that's given in blue. And the feedback then will follow that contraction and try to maintain the patch clamp seal, which we see in black. So there's only a small deviation during that contraction in the cell. If we turn off feedback, so if we now just try to measure the force and we apply a voltage pulse, we can measure the current, and we can also measure mechanical contraction, the current in black and the mechanical contraction in red, and this is the figure below. Last thing I would like to describe is chemical stimulation in tissues, really. So this is research out of the University of Potsdam in Germany. In this case, the FluidFM probe is front-loaded. This could be done if you have only a limited amount of material or high concentration of material. The pipette tip is brought in contact with the tissue, and then we deposit locally at a single cell within the tissue. Then we can just either deposit it there, or we can also track how that material diffuses through the tissue. So first, what they did is they took a dye, Cyto9 dye, and they did a global stimulation of tissue, and you can see that it lights everything up, every cell up. Now, if you took the FluidFM system, you can now target a single cell, and you can notice that only a single cell on figure C on the right is activated there. So now they know they can target single cells within a tissue with this. They also demonstrated the difference between, they actually now took this a step further. So in insect tissues, the neurotransmitter dopamine can induce intracellular calcium increases by binding to the G protein coupled receptors. So in this study, what they did is they took they took the tissue, insect tissue, and incubated it with a calcium sensitive dye. Then they took the FluidFM pipette and they stimulated the cell that's indicated in the red square in the middle image there with dopamine. So this triggered the calcium increased, and then in all the other squares around, they monitored the fluorescence there and monitored how the calcium propagated throughout the tissue, and with a couple seconds of resolution. So you can notice that in the figure on the right, you can see in the red where they activated it, first cell, that the cell across the length of that, or across the width of that tissue, activated a couple seconds after. So another thing that can benefit of using this system, especially with small volumes, the picoliter volumes of material, is that we can study short-lived radicals such as nitric oxide. So nitric oxide is a secondary messenger. It's involved in neurotransmission, immune defense, and cardiovascular homeostasis. In this case, they applied nitric oxide to a single cell in the tissue and tracked its diffusion throughout the tissue. Now, according to authors, this is the first time that nitric oxide release could be detected in salivary gland tissue. And with that, I'm going to turn the presentation over to Aran, and she's going to describe some of the work she's done with injection and extraction.
Okay, so now I would like to switch to two additional manipulations. So as we have seen, it's already so we've seen by Edward already a lot of different manipulation that can be performed on individual cells using the FluidFM. And now I would like to switch to injection and extraction. Those are two manipulations that whereby we bring the FluidFM probe now inside individual cells, and we will manipulate fluid inside the cell directly, either to inject an external solution into an individual cell, or in order to extract cellular fluid that will then further handle to perform molecular analysis on a cellular biopsy. So starting with injection, in order to come inside the cells, we are using FluidFM probe, like shown on the left, with a small pyramidal tip, whereby the aperture is on the side of the pyramidal tip, typically an aperture of hundreds of nanometers. And this is on the side of the tip, so you have really a sharp and small tips to be inserted inside the cell. And then we can come in proximity of a targeted cell and align this pyramidal tip either above the cytoplasm or the nucleus of the cell of interest. And then we can use force spectroscopy, so this very precise force control, to basically drive the probe down towards the, the cells and then insert this probe inside the cell. Once the probe is inserted, we can stabilize the probe inside the cell by keeping a constant force. And during this time, we can now use the microfluidic system to apply, in that case, a small overpressure pulse to release some solution inside the cell. And after this, you can remove the probe from the cell, also with this very precise force spectroscopy measurement. And so using the force spectroscopy for this manipulation provides us two major advantages. On the one hand, we can monitor, as Edward described before, this probe insertion inside the cells. But it also allows us to really operate the probe with a very high precision so that we can really minimize any cell damage and avoid any mechanical damage to the cell that we are manipulating. Now, on the movie here, now we can see more or less in live speed an example of such an injection in the cytoplasm of a cell, here just a fluorophore simply. And like has been more or less shown before, for extracellular release of solution, we can control the pressure pulse that we are applying to deliver solution. And in that case, inside the cell, this allows us to tune the volume that we're injecting inside cells, typically in the order of some tens or hundreds of femtoliter. And we have here a small limit, so if we go above around 1 picoliter, then the volume injected starts to be too large and to damage the cell. But as long as you remain below this about 1 picoliter injected volume, then you can really inject the solution without any damage on the cell. And this method can be used to inject different types of compounds or soluble compounds inside cells, so typically proteins or nucleic acids or any small molecules. And we have here one example of a transfection where we basically inject a mixture of a fluorescent marker together with a plasmid DNA directly into the nucleus of a cell. And you can see on the third image the fluorophore injecting in the nucleus. So this allows us to monitor the injection itself, but also to quantify the volume that was injected inside the cell. So in that case, we know we have about 1,500 copies of our plasmid. And then we can monitor the cell over several days after injection. In that case, we see the cell after 2 days that is alive and now expressing the GFP that was encoded on this plasmid DNA. So this was a very quick overview for injection, but we can also, using a similar approach, extract cellular fluid. So here we use really the same protocol to insert the probe in a non-destructive fashion inside the cell. And then instead of applying an overpressure pulse to release solution, we will now apply under pressure in order to collect some of the cellular fluid into our cantilever probe. And for this application, now we have the probe filled with mineral oil. And this allows us, on one hand, to confine the extract on the front of the probe so that it doesn't get lost into the microfluidic system. But it also allows us to monitor in real time with the optical microscope the extraction process. So you see for us on these bright field images, just nicely see the front of the extract that is filling the probe during the extraction. And then I also show here a movie with GFP expressing cells. So we have 2 cells and we are extracting the lower cells. And you can see in the movie how the cells is getting depleted in this GFP, which is in turn collected in our cantilever probe. Now, we can not only follow in real time this extraction process, but as we know the dimension of our microchannel, we can also directly quantify the extracted volume and the volume collected in our FluidFM probe. So you see on this area of picture a little bit of the range of volume that we can withdraw from individual cells. So the lower volume are in the order of hundreds femtoliter, which would typically correspond to a few percentage of the total cellular content. But we can also go up to 8 picoliter, which would fill the entire cantilever, which would be more than the volume of mammalian cells that are typically in around 2 to 4 picoliter in volume. So we can harvest either a few percent or the entire content of individual cells with this approach, and we can monitor this extraction in real time and quantify in real time the volume that we want to extract. Now, we knew from the previous work on injection that we can insert the probe gently inside the cell and withdraw it without compromising cellular viability. But in the case of extraction, of course, we are not only mechanically interacting with the cell, we are also depleting the cell from some of its content. And so we checked also cellular viability after extraction, depending on the volume that were extracted from those cells. So we see first here some data with the viability stain performed directly after extraction. So on the upper row, we have some volume distribution for cytoplasm or nucleus of HeLa cells. And in the lower part of the graph, we have in green volume extracted, whereby the cell was viable after extraction, and in red, whereby the cell were not viable. And what we saw with these studies is that both for the nucleus and the cytoplasm, it is possible to extract volume in a certain range without compromising cellular viability. So for the nucleus, we could extract up to 600 femtoliter from the nucleus with more than 80% cells surviving the extraction. And for the cytoplasm, we could even extract several picoliter from the cytoplasm of the cell with more than 80% of the cells surviving the extraction. Now, those viability stains were performed just a few hours following extraction, but we also checked with time-lapse microscopy after 1, 2, or even 3 days after the extraction whether the cells were really still alive and living. And as you can see in the movie, we have the upper cells that we didn't touch, but we extracted about 3 picoliter from the cytoplasm of the lower cells. And you can see that both cells are alive and dividing following the extraction. Now, with this, we showed that we can go inside cell, extract a small biopsies while preserving viability. But the next question was, what are we extracting, and can we really do molecular single-cell analysis with this approach? And so first, with 2 different approaches, we tested which molecules were extracted with this process. And so we use fluorescence microscopy monitoring, so labeling a few different components or molecules inside cells and checking whether they were extracting or not. And we also use transmission electron microscopy, so there we extracted cells and then redispense the extract on electron microscopy grids and perform a negative stain followed by transmission electron microscopy to image the content of those extracts. And what we saw with these 2 studies is basically all the molecules that were in the nanometer scale, so up to 2 to 300 nanometer in size, were extracted. But all the larger molecules, like small organelles or the chromosomes, or even small proteins that were bound to those larger structures, those typically in the micrometer scale were not extracted. And these results were in good correlation with the size of the aperture that we were applying. In that case, 400 nanometers, and you can really see how this aperture is basically acting as a molecular sieve, and everything that is smaller and soluble can be extracted. Everything that is larger will remain in the cell. And so after this, we wanted to show or to see whether we were able to really analyze those very small amount of molecules contained in those cellular biopsies. And at first, we looked at the transcript using or developing qPCR assays for targeted transcript from those cellular biopsies. So in there, PCR amplification allows us to afford a small dilution of those cellular biopsies. So the approach we developed for that, we basically extract cells either from the nucleus or the cytoplasm, and then we will transfer the extract, picoliter extract, into a preformed microliter droplet. So this is a very strong dilution, so it's a 1,000,000-time dilution of our cellular biopsies, but through PCR amplification later, we can really still detect transcripts. And so you see on the picture an image of the cantilever and the front of this microliter droplet. We simply insert the cantilever into this drop and then can apply overpressure to release the collected extract. And with this approach, we were able to target 3 different transcripts. So we have GFP and 2 housekeeping transcripts that we looked into the cytoplasmic and nuclear extract. So each bar here is from a different extract and different volume. And we could, in most cases, detect the 3 transcripts, even in biopsies as small as 600 femtoliters. In addition, we could also show that it's possible to both extract the cytoplasm and the nucleus from the same cell and to perform this qPCR assays in parallel from the 2 different cellular compartments. Now, after this, we wanted to also look into proteins and see whether we can also analyze proteins contains into such cellular biopsies. And there we worked on a few different enzyme activity assays. And for those, we need a reaction in liquid, but this time we cannot afford this 1,000,000-time dilution like for the transcript. So we had to come with a different approach. And for this, we use a microwell array, so small microwell that have volumes of a few picoliters, so in similar range as a cell or as our extract. And then we can pre-fill those well with desired biochemical reagents. So in our case, we use fluorogenic substrate for a specific enzyme, whereby if the enzyme is present, it will transform the substrate into a fluorescent product that we can monitor by fluorescence microscopy. And so in the images in the upper row, you can see the pre-fill microwell, then the deposition of the extract with the FluidFM probe into this microwell. And after this, on the lower row, you see the monitoring of the fluorescence that reflect the enzyme activity from the biopsy. And so with this, we developed a first assay to look into the beta-galactosidase activity from HeLa cells. And here we compared native cells to cells that were transfected to overexpress the enzyme, and we could indeed detect much higher enzymatic activity in the cell with a larger number of the enzyme. And after this, we developed another assay for caspase-3. So this time it's an enzyme at the end of the apoptotic cascade in mammalian cells. And there, in the first column, we see a few extracts analyzed from native cells that show no activity, so they are not apoptotic. And then in the 2 following columns, we see cells that were treated with a chemical that induced the apoptosis, and we can indeed detect an increase or an activity of the caspase-3 enzyme. And in the last column, we also had cells that were first infected with the virus, and then subsequently subjected to the same chemical treatment to induce the apoptosis. And there you can see that there is no enzyme activity, reflecting that the virus can inhibit this apoptosis of the host cells in order to replicate in the host. And lastly, after looking into protein and transcript, we also wanted to see whether we can analyze metabolites from those cellular biopsies. And for this, we use an approach coupled to multi-TOF mass spectrometry. And in that case, the analysis is performed on samples that are deposited on solid substrate in air or even in vacuum for the measurement. So for this, we use a standard mass spectrometry substrate, but with small particularity that we use a microstructured substrate. And this is simply so that we are able to relocate the spot on the substrate where we deposited the small extract. And so in the first picture, you see the whole array of microstructure substrate. And then we zoom in on one defined spot of this substrate where we can approach with our FluidFM probe containing an extract. We can dispense the extract locally on this spot. Of course, we can repeat this for several extracts. And after which, we can apply the multimatrix on the whole substrate and perform the mass spectrometry measurement. And so with this approach, we were able to detect and identify some 20 metabolites from HeLa cells. So those depends, of course, on the multimatrix that we use. And in that case, we could detect ribonucleotide, activated sugars, amino acids, and so on. We could even go one step further and show that it's possible to perform labeling experiment with this approach. So in that case, we compare spectra in blue from cells grown in standard medium with spectra in red from cells grown in fully labeled glucose. And there you can follow up or monitor basically the incorporation of this labeled carbon atom into the downstream metabolite that use the glucose. So typically for the ribonucleotide, we had a fully labeled ribose, 5 atom of carbon incorporated. Or for the activated sugar, we had both the ribose and the sugar labeled, for instance. So with this was a short overview on how we can go even inside cells to either bring a soluble compound to the cell or to collect cellular biopsies and then further handle those picoliter biopsies to deliver them to adequate substrate in order to couple this extraction to the downstream bioanalytics. And this is opening new ways for single-cell analysis, allowing now to avoid this single-cell isolation and lysis and sacrifice of the cell for the analysis. And so with this, I would like to give the word back to Edward for the end of this presentation.
Thank you, Iran. That was a great overview of the work you were doing. I'd just like to finish up by describing a little bit about a little more details about the microfluidic probes that we use. So there's two types of probes that you can equip with the FluidFM microfluidic system, and these are refers to the aperture size. We have probes with large apertures. These are good for mammalian cells, for manipulation, drug and chemical deposition, as I described in the earlier part of this talk. We also have small aperture probes. These are great for injection and extraction, or bacterial manipulation or lithography even. We can also customize your probes, so we can get them so it looks kind of like an injection probe there on the right. The FluidFM system integrates with your inverted optical microscopes. You have all the functionality of your optical microscope, the fluorescence, but now you have this micromanipulator with the microfluidic system. We also have the system as a standalone benchtop version. This includes the FluidFM system, the nanomanipulator, the inverted optics, and a fluorescent light source. So if you don't have the inverted optical microscope, you can get just a simple benchtop version. So in summary, FluidFM is quite a versatile tool. We've already described a number of applications, including single cell adhesion, colloidal spectroscopy or force measurements using cells, injection extraction, we saw a section on that, and on chemical stimulation using the FluidFM probe. And with that, thank you for your attention, and we will be taking your questions following this.
Okay, many thanks, Edward and Lorraine, for your presentation to us today. So we now have a little time for our questions and answer sessions. If you haven't yet submitted a question and you would like to, then now would be the time to do that. So let's go ahead and see what questions have come in so far. Our first question is, does the probe get damaged when it touches a cell or substrate, or does it damage the sample?
I'll answer this one if you don't mind. The probe is on a cantilevered spring that will bend upward when it makes contact with the cell. This reduces stress on the cell and prevents damage. In addition, the forces that are applied are very, very low, so there's very little stress there on that cell.
Okay, thanks. Second question is, can this technique be used to study nanoparticles to cell interactions?
I think I'll take this one too, if you don't mind. Yeah, it can. If the nanoparticles or clusters of nanoparticles are smaller than the diameter of the opening. So for the small standard openings, this means clusters that are less than about 500 nanometers. But we also have probes that have openings of 2, 4, or 8 microns and in diameter, and this can be used with larger particles, and this is something I talked about earlier.
Okay, thank you. The next question is to array. There's a different time axis for injection versus extraction. Can you comment on this?
Yes, sure. So it is indeed somewhat easier to push a solution out of the probe or out of the microchannel compared to withdrawing or sucking a solution into the probe. And we do so when we inject, we typically use some tens of millibars or relatively low pressure. And just within 1 or 2 seconds, we would already reach these hundreds of femtoliter delivered. But for the extraction, there we have to use much higher under pressure, so in the hundreds or up to 1,000 millibar. And it will still take 1 or 3 minutes to collect the hundreds of femtoliter or 1 picoliter. So you can tune this a little bit by making the aperture larger and having a faster flow, but we always have a much slower flow during extraction compared to injection.
Okay, thank you. Next question is, regarding force measurements, is it more like FD curve measurements using the FluidFM probe? What's the difference between regular FD measurements using colloidal or normal AFM probes and FluidFM probes?
I could take that one. So yes, it's just like doing force distance curves, FD curves. The difference is that with the FluidFM probe, if you're going to do colloidal FD curves, we can reuse the colloidal particle, or we can exchange a colloidal particle without having to change the actual cantilever or the probe. So this means you can calibrate your cantilever once and then use many different size particles doing it that way. And it's in the long run you save a little bit of money just doing that and time too. So it's similar, but we have an added benefit that we can kind of reuse the systems.
Okay, thank you. Next question is again to Irene. Although there was a clear statistical significance on the data, what is the reason for the high variability in the data obtained when comparing, for example, the cascades after different times?
Yeah, so it's a good question. So we do expect a lot of cell-to-cell variability in the amount of the enzyme, and yeah, we had to develop those fluorogenic assays at this very small scale. So I think we also have a little bit of variation on the volume we measured, and the fluorescent intensity we measured that are also relatively small. So yeah, we clearly see differences between different treatment on the cell, but we do have indeed quite a high variation. Yeah, we also have a very low amount of sample. So we compared like 4 or 5 sample of one type of cell against 4 or 5 from another type. So I guess we could reduce the variation by by collecting and analyzing more sample. Yeah.
Okay, thanks. Next question is it possible to reuse the FluidFM probe in long term? Is the injection probe only for one-time use?
I can,
Yeah,
sorry.
go ahead. Yeah, go ahead.
Yep. No, just for so we do keep a probe from one experiment from one day to another day. Usually preserve the probe in liquid, so that of course if you dry it, then you have a lot of material absorbing and potential clogging of the probe. But then it also depends the manipulation you do. So for injection or extraction, we are contacting, entering in the cell many times during a day, and after some times we damage a little bit the coating of the probe. So we have always a non-fouling coating to interact with cell, and this one is of course degrading along the usage. But we can use the probe for several days, and for adhesion measurement, for instance, we can even keep some probes for several months or so.
Okay, thank you. Next question is, how easy is it to handle the FluidFM system, including force control optimization, not to induce any physical damages against the cell?
So the system, you can set a kind of the maximum force you want to apply to a cell. You do have to know how much a cell can handle in force and in stress. So some cell types can handle a lot more force than others. But once you know that, you can set in the systems and tell the system, I only want to apply a certain amount of force, a maximum amount of force to the cell, and the system will, with the force feedback, will only apply that amount. So it's very easy. It's a single parameter that we change. We have to beforehand. We do have to calibrate this cantilever, this spring. So all of them are slightly different. They have a range of values, but there is an automated routine to calibrate those cantilevers. And after that, all it is you tell the system how much force you want to apply, and it does that.
Okay. Next question is, do we need to clean the probe after each experiment?
Oran, you want to take that one?
Yeah, I can. So, yeah, what we do is that we coat ourselves the probe with a non-fouling coating before doing any of the biology experiment. And so with this, we don't clean the probe, so we use it until the coating is not efficient anymore. So there are some ways so some coworker have used small washing in bleach or this kind of solution. I think it's not that good because it doesn't prevent the interaction between the cell and your probe, so the absorption of proteins on the surface of your probe and so on. So you may damage the cell if you don't have the coating. And it's also so short exposure to bleach also will not damage the probe, but longer exposure to some solvent or bleach that would unglue the chip of the holder of it. So we usually avoid to clean the probe.
Okay, thank you. And there's the next question or another question to the topic of the calibration. What other parameters do we need to set up for the calibration?
So for calibration, there's really only two things we have to know. One is if we bend the spring a certain distance, how much does the laser deflect? And the second thing, and so there's a routine to kind of to get that relationship. And the second is if we apply a force to the probe, how much will it bend? And so for that one, there's a separate kind of calibration step. So there's two things there. One is the deflection sensitivity, it's called, and the second thing is just the spring constant. Both of those can be done in a simple workflow, and it's maybe the first thing you do once you set up your system. And once that's done, you can run the system for as long as you want with those values. So it takes 5 to 10 minutes to do that calibration step. But after that, you can do as much experiment as you want.
Okay, thanks. Next question is, can one study quantitatively the adhesion force between a virus and a target cell? I saw data for virus injection, but I was wondering about that.
I can take this one. So this hasn't been done so far, or at least I don't know about it. In theory, so from practical point of view, it would be possible to have a large virus immobilized on a tip, on a FluidFM probe, and to come in contact with cell and to measure forces. Now I have honestly not a clear idea of which would be the range of forces that you would need to be able to measure. What I can say is that we can now measure forces down to the piconewton range, which are forces typically of bacteria to different surfaces. So we are sensitive enough for that, and so I'd say yes, it should be possible to measure interaction of a virus to a cell, but it hasn't been done so that I know.
Okay, thank you. Next question is, can you extract or inject larger objects?
Yeah. Actually, we can. So as I showed in the presentation, that the size of the object we can extract or inject is really limited by the size of the aperture of the probe. So in the case of those pyramidal tip aperture, because the channel 8 is usually around 1 micrometer, so this is not really limiting. But then the aperture is in a few hundreds of nanometer, and every object that is larger would not go through in one or the other direction. Now, as Edward mentioned at the end of the presentation, we can tune, so we can do customized probe where we use a focus ion beam to make the aperture, and there, of course, you can make the aperture smaller or larger. And with this, we were, for instance, in our group, we are able now to manipulate small organelles like mitochondria or some part of the ER. So typically going up to some 800, 900 nanometer in size. So maybe one limit is that you need the entire aperture to be inserted inside the cell. So you're limited by the age of your cell. And of course, you cannot have a larger aperture than the cell 8 to inject or extract it.
So we've come to the last question. Is the membrane not left leaky after puncturing with the probe?
Yes. So no, there it's working really well. So we know that the cell membrane is very fluidic and can easily reseal. In the case of the FluidFM approach, we also have pyramidal tips. So we are not punching a hole that is open. We are really gradually inserting a pyramid inside a cell, and the membrane stays sealed around the probe. So we do see, with working with fluorophore, during the injection, if we are inserted inside the cell, then we don't see any leakage. And then when we withdraw the probe out of the cell, we also see that the fluorescent dye is remaining either in the cytoplasm or in the nucleus. As in the example I showed for the transfection, we typically could even see the dye several days, like 3-4 days after injection in the nucleus. The dye is still there. We don't have any dye in the cytoplasm. So I think there is really no leaky problems there.
Okay, unfortunately, at this time we have run out of time, so must conclude now the question and answer session of the webinar. And so at this time, I would like to thank everyone for attending today's web seminar. Thank also to NanoServe for all their support with this event, and of course a special thanks to you, Edward and Lorraine, for your presentation to us all today. As a reminder, today's web seminar has been recorded and will be available for viewing in the day or so, and we'll send you all an email with details of how to access all of this on-demand information. In this archived version, there will be a feedback area should you have any further questions or comments that you wish to make. And so this concludes today's web seminar. And so from Edward Nelson and Lorraine Guillaume-Jonty, and from me, Birgit Washburn, it's goodbye.